Greetings, Chem students. This is Mr. Braun, and today we are doing the pre-lab video for the determination of the percentage of acetic acid in vinegar by the titration with sodium hydroxide. So basically, in layman's terms, what we're trying to do is we're trying to figure out how much acetic acid is in vinegar. What percent of vinegar is acetic acid? So you're going to need a 24 well plate. Okay, you're going to have 50 milliliters of distilled water, a rinse water cup, 1 pipette, 3 toothpicks. Now, what I would do if I were you is, well, I guess we're only using 10 drops in the well, so you do not need to stretch the pipette. Okay, so just use a normal pipette. You are going to fill that pipette with 1% acetic acid solution. This is our test solution, or excuse me, this is our known solution, so we're going to use that to compare later on. So you put in wells A1, A3, A5, you put 10 drops of 1% acetic acid solution. To those wells, you add 1 drop of phenolphthalein. So, since we have phenolphthalein with an acid, what color should you see? Yes, ladies and gentlemen, you will not see a color. It should be colorless or a very dim white, okay, since you have an acid. You're going to rinse the pipette 5 times with distilled water, and then you're going to fill the pipette with 0.200 normal sodium hydroxide. Now that N, that normality, is what we were talking about. We did it earlier on, the normality of a solution. It's dealing with the equivalence. So you're taking a 0.200 normal solution of sodium hydroxide. You're going to add the sodium hydroxide drop by drop until the phenolphthalein turns the solution a light pink color. This light pink color should persist for more than 30 seconds. Okay, so you'll add a drop of sodium hydroxide, stir it with your toothpick. Add another drop, stir it with your toothpick. Keep adding drops, counting drops until it stays a light pink color for 30 seconds. Record that number onto the data table as the standard titrant. So that is our standard. So if you look down here, here's your standard. That first one we just did would be right here, A1 standard. You do the same things for A3, the same thing for A5. Clean out your pipette. Now, we're going to do 2 different brands of vinegar, a brand A and a brand B. So what you're going to do now is you're going to place 10 drops. After you've cleaned out your pipette, you're going to place 10 drops of brand A into cells C1, C3, C5. Add phenolphthalein into C1, C3, C5, only 1 drop. And then you're going to do the same thing with the 0.200 normal sodium hydroxide. Once the color has persisted at that light pink for 30 seconds or more, you're going to record that into C1, then C3, then C5, then you'll get your average for brand A. You're going to follow the same exact procedure for brand B vinegar. So you'll get the brand B vinegar, you put 10 drops into E1, E3, E5, and you're going to come up with a brand B. And E down here would be the average of the 3 trials. Okay? To determine the percentage of acetic acid in vinegar, you use the following. The percentage of acetic acid in vinegar equals the brand titrant average divided by the standard average. So what you're going to do for brand A is you're going to take this value here that you got for the average of brand A, and then you're going to divide that by the average of the titrant. Okay, remember that titrant was 1, so that would give you a number here. And then for the final one, you're going to use brand B. So you'll take E here divided by A here to give you this answer. Okay? So, not a terribly tough lab procedure-wise. This has been Mr. Braun, the pre-lab video for the determination of the percentage of acetic acid in vinegar by titrating with sodium hydroxide. Chem students, let's investigate.